Hello everyone, and thank you for coming today. My name is Erika De Benedetti, and I will manage this webinar. Before to get started, I would like to give you a couple of information. Firstly, this webinar is recorded, and the recording will be available on biostimulant.com and on our social media in the next days. Secondly, I invite you to use the Q&A box that you can find at the bottom of your screen to submit questions. We will collect your questions and answer to at least some of them at the end of the presentation. In this webinar, we will continue to explore sustainability in crop production with the topic toward a more sustainable iron nutrition of plants from synthetic to green. It's my pleasure to introduce our speaker, Professor Youri Pii. Youri Pii is an associate professor at the Free University of Bolzano. His research activities are mainly focused on the study of the interactions between plants, soil, and microorganisms, aimed at improving the uptake and use of mineral nutrients, the physiological and molecular responses in plants subjected to variable nutrients availability and the physiological and molecular responses in plants treated with biostimulants. So, welcome, Professor Pii, and the floor is yours.
Okay, thank you for the very kind introduction. So, thanks. Before starting, I would like to thank the organizer for having me here. And as said, today we are going to discuss a little bit about iron nutrition in plants and what has been done up to now using synthetic fertilizers and what we could do for our future using, let's say, greener approaches in agriculture. Before starting discussing about these approaches, I would like to give a brief introduction about iron and iron nutrition in plants. So iron is a very abundant element on the earth crust, which is actually in which it is actually the fourth most abundant element in the lithosphere. But however, because of its chemical properties, the solubility of iron is extremely low in the majority of the agricultural soil, especially in, let's say, in alkaline condition. And in this case, the concentration, as you can see here, of iron can be very low depending on the soil pH. So this might represent, let's say, a problem for the iron nutrition actually in plants. So the most frequent or recurrent form of iron in the soil can be the iron 3+, but occasionally we can find also iron 2+. These are the main two the two main forms present in the soil solution. On the other hand, what we can say is that in case of plants, and especially in the case of dicot plants, iron is taken up as iron 2+, whereas in the case of monocot plant, it is taken up as iron 3+. So depending on the let's say on the plant species, we will have a different form of iron which is taken up. So as I said before, in the majority of agricultural, let's say in the majority of the agricultural soil, iron is, let's say, display features a very low solubility. And especially when let's say the condition of soil pH is, let's say, around let's say above the neutral pH, so below above 7, so at pH 8, for instance, which is the pH typical of alkaline soil, the iron can be very little available to plants. So if we look at this, let's say at this scheme here, basically the scheme represents the, let's say, the soluble form of iron depending on the pH, whereas up here we have the plant requirement for iron nutrition. So if we stick to the pH condition I mentioned before, so above 7, you can see that the, let's say, the total organic iron is, the availability of total organic iron is very low, whereas the, which is, I mean, the concentration available is very different from the concentration required by plants to, for an optimal nutrition. So here we have actually this, let's say, this big gap between what is available and what is required for plants. So we have this, let's say, this difference is about 4 to 5 order of magnitude in difference. So it is clear that what we can say by this, let's say, little information I've given is that in the majority of the agricultural soil, the iron is present because, as I said before, is the fourth most abundant element in the earth crust, but it is little bioavailable to plants. And being little bioavailable to plants means that plants can often suffer from iron shortage, which can lead very often to, let's say, to a nutritional deficiency, which is actually correlated with loss of production in terms of, let's say, agricultural productivity, and also decrease in the quality of the agricultural products. So if we look at plants which are, let's say, subjected to iron deficiency, we basically, at macroscopic level, we can see, let's say, symptoms which are, let's say, reduction in the content of chlorophyll of leaves, in particular in the young leaves, and also we can see some, let's say, modification at the level of the root system, in which we have, let's say, a reduction in the growth, but also we have this, let's say, growth at level of the root apexes, in which we have the production of root hairs in order to increase the surface of iron absorption. But let's say, to understand all these, let's say, changing in the plant physiology, before we have to, let's say, get a little bit more into the detail about the mechanisms that plants can adopt in order to acquire iron from the, let's say, from the soil solution or from the substrate they are growing on. So in general, as I mentioned before, we can subdivide plants in 2 big groups. So we will have plants using a strategy 1 and plants using strategy 2 for the acquisition of iron from the substrate. Strategy 1 encompasses plants belonging to the dicot and non-gramineous monocot, whereas strategy 2 encompasses plants belonging to the monocot family, to the gramineous family. So in strategy 1, basically plants adopt, let's say, a strategy which is called solubilization reduction strategy. In this case, we have that plants can release proton in the rhizosphere, so they acidify the rhizosphere soil. In this case, the acidification of rhizosphere soil will increase the solubility of iron 3 plus. This solubilized iron 3 plus is then chelated by other organic compounds released by the plant themselves. So organic acids, basically, we will have, for instance, citric acid, and also phenolic compounds. Once this iron 3 plus is complexed by this organic compound, is then reduced by this enzyme, which is called iron chelate reductase, which is able to reduce iron 3 plus to iron 2 plus. Then the iron 2 plus does dissociate from the chelating compound, and then it can be absorbed by through the action of this transporter, which is called iron regulated transport, IRT1. Whereas in the case of strategy 2 plants basically relies on the exudation of chelating compound. In this case, these chelating compounds are modified or modified amino acids belonging to the muginoic acid family. to the family of the muginoic acid. These compounds, also known as phytosiderophores, are very, let's say, are able to chelate iron 3+ with a strong affinity. And once these phytosiderophores are chelated with iron, they can be taken up by plants as a complex through this transporter, which is called yellow stripe 1 or yellow stripe-like complexes. So, so in order to, let's say, face this, let's say, limited availability of iron in the soil, plants can develop different strategies. For instance, in the case of dicot, we can, so of strategy 1 plants, we can let's say, list several strategies. As I said before, the first strategy is to induce morphological changes in the root system. And as I said before, the morphological changes basically are related to the increase of the, let's say, of the uptake surface at level of the root apexes. So basically we have the development of this intense root air system, which basically increase the surface of contact between the plant and the soil substrate. The other strategy is to increase the ability of reducing iron at root level, so the activity of the iron chelate reductase. So here in this picture, we have a comparison, for instance, of plants grown in iron sufficiency and in iron deficiency. And in the iron deficient plant, you can see that at level of the root apexes, where we have the development of the intense root air system, we have this activity, which is strongly increased compared to the iron sufficient plant. And the activity basically is monitored by a biochemical, in this case, a biochemical assay, which indicates that when we have these red colors, we have a strong reduction of iron carried out by this enzyme. Why is this activity so important? This activity is so important because, as I said before, strategy 1 plants are able to absorb iron 2+, so they need to reduce iron 3 to iron 2. And this activity is also connected by the so-called iron efficiency in plant. This is an example in which different cultivar of soybean, known to be differently resistant to the iron deficiency, are able to perform the, let's say, reduction of iron to a different extent. So the same species, soybean, different cultivar, so different variety of the same species are able to reduce iron to a different extent when they are subjected to iron deficiency. And this basically is the base of this A7 cultivar being more iron efficient respect to the other. Another strategy that plants can use in order to, let's say, cope with iron shortage in the soil substrate is to increase their ability to, let's say, acidify the rhizosphere. As we said, the acidification of the rhizosphere is, let's say, the prerequisite in order to have the solubilization of iron 3+ from insoluble soil particles. And sorry, and also in this case, as you can see here, we have a biochemical assay, let's say, which is able to localize the acidification activity. Again, iron sufficient, iron deficient plant, and the yellow coloration means that we have a strongly acidic environment. So at this level, where we have also, if you remember the previous picture, the reduction, the increased reduction of iron, we have also an activity which is able to, let's say, increase the solubilization of iron from insoluble soil particles. What is happening in strategy 2 plants? So in monocots, we said we have a different strategy. which is based on the release of these phytosiderophores, which are able to chelate iron 3+ from soil. And basically in this, in monocots, the increase in the ability of collecting iron from the soil substrate is connected to the ability of plant to release a higher content, a higher quantity, higher concentration of this phytosiderophore. Here you have an example, a field observation in which sorghum and maize are compared. And as you can see, basically the different iron tolerance between the two plants is connected to the amount, the concentration of phytosiderophores released in the case of iron deficiency. Here you have basically another, let's say, example to state the same, which is basically similar to what we have seen before in the case of iron efficiency in the dicot plants. In this case, the iron efficiency is connected to the quantity, to the concentration of phytosiderophores released by plants grown in iron deficient conditions. But this so this actually higher ability to release phytosiderophores will be connected indeed to the higher ability of the plants to synthesize the mucineic acid compound, but it is also connected to a higher ability of these plants to release this compound in the rhizosphere, and also to a higher ability of these plants to, let's say, collect the complex phytosiderophore iron from the rhizosphere environment. To this, let's say, to confirm this statement, we had some pieces of evidence in this case. For instance, you can see that this transporter, TOM1, in the root of plants grown in, let's say, iron deficiency condition is more expressed at level of gene expression. So we have also a higher amount of protein present on the cell membrane. So this actually is connected to the higher ability of the plant to release phytosiderophore in the rhizosphere. On the other hand, if we look at the, let's say, the other way, so the way back into the plant, the way back is, as we said, is mediated by this transporter, the yellow stripe 1 transporter, and also this transporter is more expressed in the root of plants grown in iron deficiency. This actually is also more expressed in the shoot of plants grown in iron deficiency, because yellow stripe 1 is not only responsible for the uptake of the iron from the soil solution, but it is also responsible for the distribution of iron at let's say at shoot level, so to the distribution of iron throughout the plant, all the organisms of the plant. So as you may imagine, the let's say the problem of iron in plant can be let's say quite important. So we need to have some strategy in order to allow plants to cope with iron shortage. And in general, the most common practice for addressing iron deficiency in plants is to use iron-based fertilizers. And actually, we can distinguish the, let's say, the conventional iron-based fertilizers in two main groups. So we can have, let's say, iron salts, as for instance, iron sulfate, and we can have also iron chelates. In the case of inorganic iron salts, they are, let's say, very cheap. And as you can see here, they are mainly used for foliar fertilization. This why is this? This is particularly important since if we apply these iron inorganic iron sources or these iron sulfates to the soil, and especially when we are working in soil substrate in which the pH is above 7, as we considered before, the iron 2 plus contained in the iron sulfate is quickly converting in iron 3 plus, and then it precipitates, forming insoluble iron oxide and hydroxides, which will not be, let's say, available to plant anymore. So in these cases, when we have to the need of carrying out a fertilization in, let's say, at soil level in, let's say, agricultural soil, which might have some problem of pH, it is more advisable to use other iron forms, which are basically the iron chelates. Iron chelates basically are compound of iron, so the iron is combined with organic compounds, which are basically belonging to the polyaminocarboxylic acid group. So as case of EDTA, EDDHA. So in this case, this organic molecule is able to stabilize the iron even at, let's say, if we can say extreme, in extreme pH conditions. So keeping the iron available to plants for a longer period of time for the absorption, for the uptake. So as I mentioned before, the most common iron chelates which are used in agriculture up to now basically are the iron EDTA, the iron DTPA, and the iron EDDHA, even though other, let's say, chelating compound are have been developed and are also being developed in this moment, displaying different and improved characteristics respect to the, let's say, respect to the more traditional one. So the problem with this chelated compound of iron is that they, for instance, as I stated in this slide, that they display a variable stability depending on the soil pH. So for instance, the example here I report here is for the iron EDTA, it is stable until pH 6. Over this pH, basically the complex is not stable anymore. And very often we may assist a ligand exchange reaction, for instance, with calcium, which can display a higher affinity for the EDTA. The same basically apply for the iron DTPA, which is stable until pH 7. Let's say the chelated compound which display, let's say, the wider range of stability is the iron EDDHA, which is stable until pH 11. Difference between the other, it is that this last one is the most expensive. Okay, so in general, iron chelated form of iron has been used, are used in agriculture because they have several advantages. The advantages is that actually that, for instance, they are required in low quantity. They are, let's say, effective in, let's say, making the iron available to for the plant uptake. They are compatible with other agrochemicals, as for instance with other fertilizer or pesticide. And actually, if we talk about iron EDDHA, we can say that under alkaline condition, they can provide a good option or a good practice for to provide iron to the plant. Actually, beside the advantages, we have also disadvantages. And actually, we are going to discuss these disadvantages in a little bit more detail in the next slide, but just to list them. Something already spoiled. I mean, the effectiveness of these chelates is dependent on soil pH, so we cannot apply the same chelated compound in every condition. The stability of chelates is influenced by the presence of other cations. Chelateds are basically molecules which are able to interact with metals like iron, but also they can interact with heavy metals. So we can have the mobilization, the unwanted, the undesired mobilization of heavy metals. And also this chelating compound being basically xenobiotic, they can be very persistent in the soil plant system and so can affect plant physiology and the quality of agricultural product. So going straight to the, let's say, to the first disadvantage in the use of this chelating compound, as we said, and we already basically discussed a little bit, is that they can display differential stability depending on the soil pH. So here you see, let's say, a drawing, a scheme representing the total chelated iron depending on the soil pH. As you can see, what we have discussed, for instance, EDTA, DTPA, in this case, the concentration of iron declines, the chelated concentration of iron decline quite steeply when we go from pH, for instance, 6.5 to 8, as well as in the case of DTPA, whereas the only stable in all the concentration or in all the pH is EDDHA complex for iron. This actually could not be a problem when we are discussing about, for instance, plants growing in a soilless system or hydroponics, in which let's say the pH of the substrate can be controlled quite easily. So we can also use, let's say, less expensive chelated form of iron like EDTA or DTPA. Whereas when we are in, let's say, in alkaline soil, in a real alkaline soil, let's say the most advised form of iron to be used in order to be sure that to maintain the iron in an available form would be the iron EDDHA, but as we said before, these represent the most, let's say, the most expensive fertilizer among the different chelating compounds. The other aspect I would also like to discuss during this seminar is, as I said, the stability of the complexes depending on the concentration of other cation in the soil solution. As you can here, we have a couple of numbers representing the stability constant of the complexes, in this case between iron 3+ and EDTA. So here we have a stability constant of 25.1, whereas this is a clear example of a stability constant of calcium complex with EDTA, which is 10.7. So this actually the consideration of the, let's say, the comparison of these two stability constants should tell us that let's say the complex between iron and EDTA should be the most stable one. So I mean, we could be, let's say, quite sure and safe in saying that the complex between iron and EDTA is more stable than the complex between calcium and EDTA. The problem is that when we consider the speciation of iron and these complexes, we should not consider only the stability constant, but we should also consider the real concentration of the element in the soil and in the soil solution, because the speciation basically is determined by the combination of the concentration and the stability constant. So if we consider calcareous soil, but in neutral pH condition, in general, we have this, let's say, very different concentration of iron and calcium. So despite having, let's say, a very different stability constant between the two complexes, the very high concentration of calcium in the, let's say, in the soil solution, in the substrate, makes the, let's say, the combination, the combination between calcium and EDTA the most favorite, the most favorable reaction. So it would be, it will be very easy that we can have, let's say, ligand exchange reaction. So the EDTA will release iron and will complex calcium in these conditions. So the result will be that calcium will be complex, whereas iron will be released as iron 3+ in the soil solution. And as we said before, when we were talking about the possibility of using iron sulfate as fertilizer for, let's say, at root level, the free iron will precipitate, forming insoluble hydroxide and oxide. So this could be a problem that should be considered when we plan fertilization plan, considering the iron nutrition. Another important aspect we should consider is the competition with another cation, which is copper. Copper actually is, let's say, an essential nutrient for plants. It is essential, but let's say the concentration range of copper within plants should be within a narrow range, because it will be very overcoming, let's say, the maximum level of copper in plants. This will, let's say, generate actually toxicity phenomenon in plants. Several pieces of research carried out by a research group at Boku University in Wien have basically established that, fertilizing plant with iron EDDHA, they basically studied the speciation of iron EDDHA in different kind of soils. They monitor the, let's say, the concentration of EDDHA, of iron EDDHA in the soil solution for a certain period of time, and they were able to see that this the concentration of this chelated compound basically decreased very quickly within the soil solution. This, they were able to establish that the decrease of this, let's say, of the concentration of this chelated compound were not related to the activity of plants. So they basically hypothesized that something chemical could happen, and in particular, they hypothesized that some other cations could be able to displace iron from the EDDHA to bind basically at its place. And actually the most, let's say, the candidate for this role was actually copper. Because copper display a very high affinity for EDDHA. And so, the high concentration of copper, both in the solid phase of soil, but also in the soil solution, could displace iron from the EDDHA. So in this case, we have the formation of this new complex. So again, releasing iron in the soil solution, which could be immobilized as in the solid phase. So not making it not available to plants, and substituting, so forming this new form of chelated compound. Actually, this is quite a complex equilibrium, but the main drivers of this equilibrium are actually the iron concentration in the soil solution and the affinity of iron of the copper concentration in the soil solution and the affinity of copper EDDHA for the soil particles. So the higher will be this concentration here, and the higher will be this process, or I mean the absorption of copper EDDHA on the soil particles, the more this equilibrium would be, let's say, in this direction. And so we will also have the release and immobilization of iron on the soil particles. This could be a problem. This could be a problem because we might say that this is another example of similar to what we have seen before concerning EDTA and iron and calcium. But this could be particularly important nowadays, considering that we are basically, let's say, observing an increasing accumulation of copper in the soil. This is actually an example. This is a map representing the, let's say, the average concentration of copper in the soil, the agricultural soils in Europe. And as you can see here in Italy, we have, for instance, let's say we may say the highest in general concentration of copper in our soil. These are actually let's say related to the location, to the presence of vineyard. And this is because basically copper is widely used in viticulture, but also in general in agriculture as fungicide, as pesticide. So again, so it is used against fungi, against pests. So this could be a problem in this case for, let's say, if we consider viticulture, but this could be also a problem in the organic farming, in which copper, for instance, is only basically is the only pesticide admitted. So this could have basically, as I said before, a double result. The first one is that we, by increasing the concentration of copper in the soil and soil solution, we are, let's say, reducing the effectiveness of iron chelated form as fertilizer. But on the other hand, we have that the chelation of iron with EDTA or EDDHA could improve or could increase the bioavailability of this metal to the plants. And as I said before, a too high availability of copper might lead to the production of copper toxicity in plants. So this could be also another important thing to consider in this case. The other aspect I would like to discuss is that chelating compound can, let's say, remove heavy metal. This is actually, let's say, this is actually what has been commonly used in the soil remediation strategies. So when we have soil which are, let's say, contaminated with the heavy metal, they can be washed with this chelating compound in order to remove the heavy metal. But this chelating compound can be used also to increase the availability of this heavy metal in the phytoremediation strategy. So in order to increase the bioavailability of this heavy metal that can be removed by plant absorption. So here I report a study very quickly, very briefly, in which a chelating compound, which is this relative of EDTA, is used to remove lead from soil. As you can see here, in the contaminated soil, we have an increased removal of lead by increasing the amount of chelating compound added. And in this specific condition, so in this, with the addition, with the supplementation of 0.5 gram per kilogram of chelating compound, these researchers were able to see that this, let's say, this chelating compound were able to increase the bioavailability of lead to plants, which, as you can see here, accumulated a very high level of lead in their tissues. And actually, not all the chelating compound has the same ability to remove this heavy metal. But as you can see here, the DTA let's say, between the different chelating compound considered is the most, let's say, effective, whereas the EDDHA could be the less effective. But in any case, the take-home message of this, let's say, of these slides should be that whenever we, let's say, add chelating compound to our agricultural system as fertilizer for iron, for instance, we, let's say, add chelating compound that could remove heavy metal if we have heavy metal in high concentration in the soil that can be made available to the plant. Last but not least, as I said before, chelating compounds are xenobiotic, so yeah, they are synthetic compounds, and they can be persistent in the plant soil system. In particular, this is an experiment showing that tomato plants grown in the presence of iron EDDHA display the accumulation of EDDHA in the leaf tissue. This is the, let's say, an HPLC analysis of pure EDDHA. Here you have the leaf extract of tomato grown on EDDHA. So here you see that you have, let's say, distinctive peaks of EDDHA in the tomato extract, whereas in the tomato plants grown on iron inorganic salt, you basically lack these, let's say, peaks. These demonstrate that EDDHA is absorbed from the soil, translocated into the plant tissues. This might have actually, as you can see here, an effect on the plant physiology. In fact, in this experiment, these researchers were able to demonstrate that plants grown on EDDHA display a lower level of lycopene, which is a nutraceutical compound very important for tomatoes. Plus, the same experiment carried out on cucumber plants show that plants grown on this chelated compound were susceptible to the infection with this pathogen, whereas plants grown on the inorganic sources were resistant. So, considering all these observation and feature, the question is now, can we find, let's say, a greener way to supply iron to plants, even though they are grown in difficult condition in difficult soil? And this question basically posed our attention on the possibility of using biostimulants as iron biochelates. So the possibility of using this group of molecules which are now attracting the attention of the agricultural sector as possible mediator for iron nutrition. So I'm going very quickly on this actually definition, because I know that in this cycle of webinar, this let's say topic has been discussed several times, but in general we can say that biostimulants are a group of molecules which are able to improve crop productivity and quality through different, let's say, through different ways, like increasing the availability of nutrients, increasing the plant nutrient use efficiency, and other kind of action. What we can say is that plant biostimulant is a very heterogeneous group of molecules which encompasses a broad spectrum of substances. All these substances can exert, let's say, a beneficial effect of plants, even though sometimes the real mode of action is still not clarified yet. Let's say, to the purpose of this seminar, I decided to focus on three, let's say, groups of these biostimulants, which are the beneficial microorganisms, the humic substances, and the protein hydrolysates. So in the case of beneficial microorganisms, I think that we all know that beneficial microorganisms are microorganisms known also as plant growth promoting rhizobacteria, which are able to establish a relationship with plants, and they are able to carry out positive effects through different kind of action like biocontrol, root growth promotion, and biofertilization. In particular, in this case, we are interested in those in biofertilizers bacteria. And in particular, I carried out a research concerning the role of Azospirillum brasilense, which is known as a nitrogen fixing bacteria, in improving iron nutrition on cucumber plants. Here you see basically you have the same biochemical assay I showed you before, so the reduction and the acidification of the rhizosphere. And as you can see, whenever you have Azospirillum brasilense inoculated in the plants were able to activate to a greater extent this biochemical activity, suggesting that the presence of the PGPR could improve the ability of plants to take up iron from the soil solution, from the soil substrate. This actually was translated in this case in a higher uptake of iron. So despite you are dealing with iron sufficient or iron deficient plant, in the presence of Azospirillum brasilense, you have more or less a 4 times higher uptake of iron. So in this case could be an indirect effect of the Azospirillum on the plants. So the plants were more ready or more active in taking up iron. But we cannot exclude that also the molecule released or the molecule present in the rhizosphere could have a role. In fact, this is, let's say, the aminoacidic profile of the rhizosphere soil in the presence of Azospirillum brasilense. And as you can see here, despite independent from the iron nutrition, we have, for instance, the increase of several amino acids, like for instance, glutamic acid, which could have a role in chelating iron from the soil solution. So we cannot exclude that also molecule mediators released by the Azospirillum could have a role in improving the iron nutrition. So this was just a brief example. The second group of substances I would like to discuss are the humic substances. So the humic substances, we know that are organic molecule belonging to the non-living component of the soil organic matter. And in particular, they are, let's say, formed of two big classes of compounds, the humic acids and the fulvic acids. We know that, we already know that they have an effect on plants' growth. So they enhance plants' growth, most likely through, let's say, enoxin-like activity, so they act like hormones on plant. But we also know that they are able to increase the release of protons, so they are able to induce a higher acidification of the rhizosphere soil. This release of proton is very important for plant nutrition, because the uptake of a lot of mineral nutrients is mediated by secondary active transport, so through the exploitation of a protein ingredient formed by the H+ ATPases at soil level. What we also know is that humic substances, humic acid, can also chelate iron, so they are able to mobilize iron from the insoluble iron sources, making it available to the plant. This actually mechanism has been demonstrated both for strategy 1 plants. So in this case, the iron is chelated directly by the humic substances, and then let's say made available to the iron chelate reductase. Whereas in the case of monocot, the iron is chelated by the humic substances, and then we have an exchange, a ligand exchange reaction. So the iron is given, is passed on to the phytosterophores and then taken up by the plant. very quick example and very, let's say, clear example. In this experiment, cucumber plants grown in iron deficiency were then resupplied with different iron sources. And as you can see here, the iron complex with humic substance was actually, it seemed actually the most efficient in releasing iron to the plant, because as you can see here, let's say these leaves were those, let's say, showing the most, let's say, the most evident reverting of the iron deficiency symptoms. Last but not least, protein hydrolysates. So the protein hydrolysates, I guess they are already clear, but in general, they can be described as a mixture of polypeptides and oligopeptides that are obtained by different sources. In general, we can have animal sources and plant origin. And in general, those obtained from the animal origin are generally produced by a chemical hydrolysis, whereas those obtained from plants are generally obtained by an enzymatic hydrolysis. So the idea of possibly using the protein hydrolysates as iron chelating compounds derived by the fact that there is a growing body of evidence showing that the iron nutrition in plants can be controlled by signaling peptide, like those belonging to the Ironman family. And actually, the protein hydrolysates can contain amino acidic residues like glutamic acid or aspartic acid, as I said before, that can be active in chelating the iron. So for this reason, we decided to undertake several experiments in order to understand whether these chelated iron biochelates could be effective sources of iron. The first experiment was carried out on both, let's say, tomato and cucumber. And in this case, these plants were grown in, let's say, in hydroponic, with a limited availability of iron, and were supplied with protein hydrolysate without iron. So just to understand whether the polypeptide, the peptides could function as signal molecule to, let's say, activate the iron uptake system in plants. And as you can see here, basically the plants treated with protein hydrolysate display a higher accumulation of iron in leaves. So mostly the concentration of iron doubled respect to the to the untreated plants. So this actually was a good starting point to, let's say, to start with our experiment. The second trial we carried out at the University of Tuscia is, let's say, a trial concerning the production of tomato in alkaline conditions. So the substrate in this case was alkaline, the pH was 8.1, and the plants were treated with different sources of iron. So we have the traditional iron chelate, the EDDHA, and then we have the peptide complexes at two different concentrations. The let's say the very nice results was that the yield of tomato plants were not different whether they were treated with EDDHA or the protein hydrolysates, suggesting that the peptide, the iron chelated in the biochelated, was as effective as the synthetic one. And actually, when we look at the SPAD value, which basically display the concentration, the relative concentration of chlorophyll in leaves, and also the iron concentration, the plants treated with the iron biochelate displayed, let's say, perform better compared to the plants treated with iron EDDHA. The same result, more or less the same result, were also obtained in a trial carried out at pH 6, the same tomato trial. But in this case, the most, let's say, important aspect to underline is that in the case of iron concentration in the tomato leaves, when we supplemented plants with 30% less iron compared to the EDDHA, we had more or less the same concentration of iron in the leaves, suggesting that this biochelate could be also a more efficient source of iron as compared to the EDDHA. Last, at the University of Bolzano, we carried out some trial in hydroponics in order to understand whether the biochelate compared to other traditional, let's say, fertilizer like the DTPA, EDDHA, and iron sulfate can have different performances at pH 7 and at pH 6 and pH 8. And in particular, here we could not see any difference at level of the biomass, both at shoot and root level. But what we were interested in is in the activation of the, let's say, biochemical activities underpinning the iron acquisition. And as you can see, basically here, especially in the assay carried out at pH 6, that the biochelate basically perform as well as EDDHA and iron sulfate in the soil solution. Whereas at pH 8, we have to, let's say, to look at this data a little bit more into the detail, because we had also plants that were suffering a little bit from the extreme pH condition. What I would like to point out from this experiment is that only 1 week of treatment with biochelate basically were very strong in reverting the let's say the iron deficiency look that we have the plants grown in iron deficiency. So this could be a very important aspect to be considered when we let's say plan or we want to develop a fertilizer based on protein hydrolysates chelating iron. So I promise I am done. So to conclude, I would say that plant biostimulant, and in particular plant protein hydrolysates, are, let's say, acquiring a bigger and increasing importance in the agricultural sector, as let's say demonstrated by the data about the market. But what I would like to say is that we already know that protein hydrolysates have a very strong effect on plants, so they are able to increase, they are able to induce positive effect on plants, like increasing yield, increasing quality. So by using the combination of iron and plant and protein hydrolysates, we could, let's say, have a very positive effect on the plant. On the other hand, what I would like also to let's say to point out is that this molecule, these protein hydrolysates, are basically biological molecule, so organic molecule. So we should not be, let's say, concerned of carrying out repeated application of this iron biochelate, even when we are growing plants in extreme pH condition. This is because these protein hydrolysates can be easily biodegraded by soil microorganisms. And on the other hand, so being easily biodegraded, we will not have the undesired problem of persistence in the plant soil system, and also, for instance, the mobilization of heavy metal. And on the other hand, being degraded by soil microorganisms, this could also be a way of selecting a positive and a beneficial microbial community, which could further improve the growth of crops. So here I'm done, and I'm sorry if I was a little bit longer than foreseen, and I would like to thank for your attention, and I would be glad to answer questions whether there are some. Thank you.
So thank you very much, Professor Pii, for this insightful presentation. As you anticipated, now we have a few minutes for a Q&A section. I just want to remind to all of you that you can still submit questions using the Q&A box that is at the bottom of the screen. We have already received some questions, so I'll start immediately with the first one. So thank you, Professor Pii, for this nice presentation. And besides those already mentioned in the presentation, are there any other possible positive aspects in supplying plants with iron chelated to protein hydrolysates?
Okay, so I actually briefly mentioned this opportunity in this slide here. In this slide, basically, I focused on the aspect of crop yield and quality. So increasing, for instance, let's say the biomass and the qualitative trait of the plants. But what we should also consider is the fact that how can I say? Is the fact that a lot of, let's say, biostimulant or protein hydrolysates has been characterized to, let's say, to improve the characteristics of plants to resist to abiotic stresses, like, for instance, drought, salinity. So actually this, the use of biostimulant as chelator for iron could also allow the, let's say, the exploitation if I can say, the exploitation of marginal lands, maybe the, let's say, the more degraded soil, I would say a little bit more degraded soil, or characterized by, for instance, by a higher salinity, where normally plants are not thriving so well. So by applying this plant biostimulant or protein hydrolysates, we might also have this positive, if we say, side effect in increasing the resistance of plants against stresses, abiotic stresses, but also maybe other kind of stresses. Plus, being, let's say, formed by proteins, the protein hydrolysates can also be a nice and useful sources of nitrogen for plants. So this could be another positive effect, a side effect we could envisage when we plan some kind of fertilization based on biochelates, for instance. Have I answered?
Yeah, I think so. Okay, so I go to the next question. Again, thank you, Professor Pii, for the presentation of today. And do you think that supplying plants with iron biochelate might improve iron uptake efficiency in plants?
Okay, this could be somehow connected or related to the answer I have just given, because there are several pieces of evidence in the literature saying that, or demonstrating that, iron nutrition and nitrogen nutrition are strictly connected. So this means that when we have plants stressed for nitrogen nutrition, so grown in the, let's say, in low concentration of nitrogen, also the iron nutrition could be, let's say, hindered or prevented. So the answer would be yes, because on one side, if we, let's say, apply iron as biochelates as we have seen, actually we have seen in this, in the result of this experiment. So when we basically supply plants with iron, which is in a concentration that is 30% less respect to a traditional fertilizer, but we are able to detect more or less the same concentration of iron in the leaves, this would mean that the peptides, the chelated, the biochelated iron could be more efficient in, let's say, giving iron to the plant. So the ability of plant to use iron would be increased. Plus, as I said before, if the peptide is also sources of nitrogen, so that the plant is well nourished from the nitrogen point of view, then it would be the iron nutrition would also be improved.
Okay, thank you very much. We have another interesting question, and that is, is it possible to enhance, further enhance the plant iron uptake by combining biochelates and microorganisms able to solubilize iron?
This is a very interesting question, and this is actually what I would like to, let's say, I would like to do in the future, to combine, for instance, the experiment with Azospirillum brasilense, which is known to potentiate or to enhance the ability of plants to take up iron, and the ability and the possibility of giving iron through biochelates. The only thing the only drawback or possible drawback in this approach could be the fact that, as I said before, peptides used for to chelate iron can be a substrate that could be degraded by microorganisms. So, this could be something to fine tune, because if we supply the biochelates and the microorganisms at the same time in the soil, and the microorganisms basically could degrade the peptide before the plants could have absorbed the iron, then we will have the problem of the precipitation of iron in the substrate. But indeed, this could be a very interesting aspect to be addressed, because as we know, when we are, let's say, carrying out an experiment to assess all these features for the biochelates, we are, let's say, in more or less controlled condition, hydroponic or soilless condition, in which, let's say, the occurrence of microbiota associated to the root is very low. But the situation might be very different in a natural soil, in which plants are strongly associated with bacteria at the rhizosphere level. So indeed, in order to assess the, let's say, the effectiveness of this biochelate, would be necessary to go from the, let's say, a controlled system to a natural system.
Okay, very good. And we have seen that the presentation was focused on iron, but we have a question that is, can we expect similar benefits of biochelates with other micronutrients?
I guess yes. I guess yes, because I mean, other micronutrients that might be provided through, let's say, these biochelation strategies could be other cations like, I mean, copper, zinc, manganese, but actually, in particular, as I explained during the presentation, for instance, copper need to be very little available to plants because it's a micronutrient which is required in very little concentration and in a very, let's say, controlled range. The other micronutrients I can think of is the zinc, but as far as I know, we don't have big issues of zinc bioavailability at the, let's say, at level of soil. So, I mean, I'm aware that in some cases we might have zinc deficiency, but very often the zinc deficiency, like other micronutrient deficiencies, are not strongly related to the lack of zinc in the substrate or low bioavailability of zinc in the substrate, but rather than with the competition of other cations for the absorption. Because you have to consider that, for instance, in the case of divalent cation like zinc, copper, manganese, all these, also iron, cadmium, they have all their own transport system, but they can also be absorbed by, let's say, in a non-controlled way through channels and other transporters. So whenever we have let's say, an excess of one cation, this excess, this means that this cation will be preferentially taken up by the plant. And on the other hand, all the other will be taken up to a lower extent. So this could be let's say, the reason why we can observe deficiency in other cations. I mean, indeed, let's say the development of biochelated form of zinc could be an option that we could look at, but as far as I know, I don't think that zinc nutrition or zinc availability could be a big problem at level of agricultural soil. But this would be to check, actually.
Okay, very good. I think we can take just a few more minutes for 2 more questions, and then we can end this webinar. We have a question about air temperature. So, I mean, if is it important to apply these biostimulants as biochelates in a particular air temperature, or if you have some experience or knowledge on the relation between temperature and biochelates application?
I mean, actually, all the experiment we have carried out in this, and I have up to now, and I have shown here, are let's say biochelates given or in the, let's say, in the soil in the nutrient solution in the case of the hydroponic system, or as in the case of this tomato experiment, as fertirrigation way. Okay, so in this case, actually, I'm not aware whether we should have checked the air temperature or the temperature. In general, what I know is that when you perform a foliar fertilization, the plants should be in a suitable water state, because let's say the fertilizer you apply, whether it is, let's say, an inorganic fertilizer, for instance, as the example we have done with iron sulfate, or in the case you spray biostimulants, because also biostimulants can be applied by foliar spray. In that case, you should have, as I said, plant let's say, in a suitable water status in order to have the channels and stomata, let's say, open in order to allow the entrance of this fertilizer within the leaf mesophyll. Okay, so this actually being necessary to have the plants in a certain water status, actually, I would say that you should avoid very hot climate or very dry seasons, I guess.
Okay, thank you. And just to conclude, a general question about iron availability. So the question is if iron-related issues are most related to iron insolubility in the soil, or is iron natural deficient in the soil in general?
So, I mean, as I said at the very beginning, iron is very abundant in the soil, actually. So the issues concerning the iron deficiency in plants is mostly related to the iron bioavailability to the plant. So this means that the majority, may I go back to the slide? Here it is. So this actually is what I was trying to explain now. So basically, the iron in general is very present in the substrate, in the soil. But what is, let's say, the problem is that despite the pH you're looking at, so the total soluble inorganic iron, which is the total soluble inorganic iron, is the iron which is, let's say, available for the plant absorption, is here. So you have this big difference in, also here, you have a difference between what is available and what is required by the plant. So the problem is not that we do not have iron in the soil, but the problem is that what is available to the plant. Okay, so the problem with iron is not the, let's say, not the concentration in the soil itself, but the available concentration to the plant, which is dependent on the solubility that is determined by soil pH and also, I mean, the redox status of the soil. So how much oxygen do we have in the soil and other chemical and physical parameters like that.
Well, thank you very much for your answer. And as I promised, it was the last question for the webinar of today. And I would like to thank you again, Professor Pii, for the insightful presentation. And I also would like to thank everybody for attending the webinar. If you still have questions, please don't hesitate to submit them on biostimulant.com website, and our scientific committee will get back to you with the answers. I would like to remind that the recorded version of this webinar will be available soon on biostimulant.com website and also on our social media. Have a nice day, everybody, and we hope to see you on the next webinar. Bye-bye.